I'm going to go ahead and play this movie maybe one more time. I think I can manage to play it again, because the question for today is understanding how you go from a few cells, this is zebrafish cells on top of a yolk, engaging in a process like this at the beginning and then more complicated things later, and somehow you end up with this at the end. That's the big picture question, like how does that happen? And before I go too far, I really want to highlight the people who did the work. So this is the product of lots of scientists working together, especially junior folks. So Ojan, Varda, Sajad, Tyler, and Indrajit are the younger folks, and some really excellent older folks too, and I'll highlight them when they come up. Great. So the big question, as I said before, is how do you turn a blob? And here the blob, the cells that are going to become the animal are actually this cell sitting on top of a yolk. And I'll show you a picture from a long time ago of how that happens. So it starts out with 4 cells, and the cells divide and divide and divide. And then there's this magical moment where the cells start to flow across the yolk. You see it? And you can start to see that an animal is really forming there. You'll start to see a tail, and here's an eye, right? And you can see the structure of the animal forms after this beautiful flow that starts, and then you end up with something that's the shape of a fish. Great. And you want to know, there's many things you might want to know about that process, but one thing you might want to know is how does it get that shape, okay? And, you know, we actually have an answer for this in a different context. So if I imagine having a glass blob, and I have a very skilled glass blower, well, that person uses a mixture of tools that apply forces and heat in order to generate something that really is the shape of a fish, right? And actually, this is my son's fish. Like, he's obsessed with fish. And I went on a trip and I got him a glass fish from Venice, hoping that would be enough. But no, now I have pet fish that are real and can wag their tail. But this is an amazing result. Okay, how did the glass blower do that? Maybe we could gain some insight from that. And how the glass blower did it is, first, you have to be able to apply maybe some localized forces, right? You apply forces to specific places to pull on the material to draw things out. And these, I might refer to them as pressures or shear stresses, where instead of, you know, a pressure where you compress something, but also you can shear something like this. Those are types of forces. And another important ingredient is the heat, right? So we know that what this is doing for a glass is it's altering the material properties, things like the elasticity of the material or the viscosity of the material. And actually, for making a fish, it's really important that the system goes through what's called a glass transition from a solid, and then it sort of melts a little bit into a fluid and then into a solid again, because that allows you to pull things into shapes and then freeze them in place so that you have like a stable morphology at the end. And so it's this combination of forces and control over the local material properties that's really necessary, I would argue, to get a specific shape. Okay, and so for the talk today, I'm going to focus on this first, the material properties first. How do organisms control their material properties? And if I have some time at the end, I'll try to get back to there. And that's going to be sort of the structure. And so maybe you can ask the question, well, are the local materials properties changing in like an embryo or this zebrafish that I talked about? So again, this is that movie where now you can see individual cells that have been labeled. And I would articulate that during this magical flowing process, you see that this behavior, I mean, even looking at it by eye, it really looks like a flow, like a fluid flow. And that when you start to see there's also a compression here that starts to, it's called convergent extension, where you start to actually form maybe what you can see as the sort of spine or notochord of this animal, and that those sort of flows in the material might be important. You say, well, maybe so. It looks like there might be some changes to the material properties going on. So now you want to do maybe some scientific work, and I have examples of a couple of scientific papers over the past 5 years that make, I would say, a very strong case that there is a fluid to solid or rigid to fluid, basically, transition going on during development in these embryos. So this is a particular paper where it shows that when you saw that the body of the zebrafish was sort of occurring and sort of elongating, during the process where that tail is elongating, there's actually a gradient in rigidity. So as you get closer to the head, it's more solid-like, and as you get closer to the tail, it's more fluid-like. And you can measure that quantitatively by looking at sort of the rate at which cells exchange neighbors, you can really quantify that carefully. And similarly, this shape kind of maybe looks familiar from that first movie I showed. This is the yolk, and this is the ball of cells sitting on top of the yolk. And right at that moment, when this ball of cells started flowing down the sides of the embryo, there's this really abrupt transition. You can prove now with modern techniques between it's rigid, it's basically behaving like a solid here, and then it's turning into a fluid so that it can flow. And that makes a lot of sense, right? If you're thinking about, I need to change the shape of something, if you're the glass blower, you say, okay, it would be stupid if I tried to change the shape into the fish when the thing was still a really hard glass, right? That would take a lot of energy and work, and that would be really difficult, and I might not get the shape I want. But I really need to convert that system into a fluid to be able to deform it in the way I want, and then I need to freeze it in place. So it also makes sense that this should be going on. Okay. So then, the other thing I want to tell you is that I talked about systems that were groups of cells together, but it turns out that inside your body there's a lot of tissues that aren't composed of cells, things like your bones, your cartilage, that are they're called acellularized. They're a material that cells excrete that then they use to make structural components. And interestingly, a lot of those components, some of those types of things are called extracellular matrix, and a lot of them actually also go from floppy to rigid, and they do so either by changing the concentration of the polymer that makes up these networks, or actually, and this is the cool part, deforming the boundaries. So if it turns out that if I take a chunk of material that makes up extracellular matrix in your body and I stretch it, it starts out really floppy, but there's a critical shape of the box at which the system becomes rigid again. Okay? And so that's another type of floppy to rigid transition we might want to understand, and it's also important, very important in biology. Okay, so also biology tells us this fun fact. Okay, I am interested in the behavior. So this is a scale. So this is meters, and these are pictures of things, right? And so one of the neat things about biology is it goes all the way from the scale of, individual components of DNA, all the way up to the size of an organism. And this is a logarithmic scale, right? So this is amazing. And the key thing is that, of course, the control systems for these processes are in your genes, right? And yet we need to articulate a fluid to solid transition at the scale of organs or the body of an organism. So that's kind of a hard and difficult problem. How do I execute that control? Okay. So biologists and maybe some biophysicists want to know, how does it do it? Like, how do organisms manage this seemingly difficult task? And material scientists, I sometimes think of myself as a material scientist too, I'd like to make materials that can do that. That would be really cool and useful as well. Okay, so we want to do these things. We want to understand these things. So now I'm going to say why I think doing these things is difficult. Okay. So, and I'll talk about it in terms of two challenges. The first challenge is that, and I'm going to explain what I mean by this in just one slide. I'll call it emergent rigidity is a difficult problem, okay? If we want to understand how something goes from floppy to rigid, it turns out it's really hard. Okay, and the second challenge is that in these systems, there are many tunable parameters, and the tuning parameters that I have control over are all so much smaller than the emergent behavior I want to control. So how do I control? Okay, so we're going to tackle challenge one first. Okay, emergent rigidity is hard to understand. Okay, and so this is a quote like, I grew up as David said, as a condensed matter physicist, and so this was a famous condensed matter physicist. He said, we're so accustomed to the rigidity of solid bodies. The idea, for instance, that when we move one end of a ruler, or a pointer, that the other end moves the same distance, that we do not accept its almost miraculous nature, that it is an emergent property not contained in the simple laws of physics, although it's a consequence of it. So I guess what I'm saying is like, yeah, we all know that this happens, but understanding why is actually really hard. And as a follow-up, Phil Anderson said, maybe the deepest and most interesting unsolved problem in solid-state theory, you know, the condensed matter, is the theory of the nature of the glass and the glass transition, because glasses are disordered. And so if you ask how this happens in a disordered system, it's even harder than if it's in an ordered system. And so this is the picture that I wanted to show you, is because, like, actually, we don't even know how this happens, quite frankly. And it's a stupid glass fish. So how can we have a hope of understanding how it happens in a real fish? I don't know. But I'll try to convince you that I think we're making progress today. Okay, so we're going to work on this challenge 1. And I'm going to tell you what the answer is, and then I'm going to explain it. Okay, and this is lots and lots of hard work over years and years that I'm sharing in a short talk. The answer to challenge 1 is there are multiple physical mechanisms that can drive rigidity in tissues. So this is what's called a phase diagram, and it says, okay, if I vary this parameter, and this one is one maybe you're familiar with, it's the density. So if I have a bunch of objects and I sort of shrink the box that those objects are living in, that's one way of changing the density. It's applying a pressure, right? And you know that if I handed you a glass of water and I said, make that glass of water solid, well, probably the first thing you'd think to do is stick it in a freezer, right? Change the temperature, change the fluctuations. That's this axis. So in a normal system, it would just be temperature, but in biology has access to active fluctuations, so it might not just be the temperature, other sources of fluctuations. So if I have a very high amount of fluctuations, I expect I'll be a fluid, and that's this white space out here. And then if I lower the number of fluctuations, eventually I expect I'll cross a boundary and then I'll become a solid, and that's this gray space here. Okay? And so I sort of have a picture of it. It's hard to draw temperature, but the idea is that when I have more fluctuations, things have dynamical trajectories that move around a lot. That's what that picture is. And if I said, I need you to turn this glass of water into a solid, but I said you can't use a freezer, you can't use temperature, maybe you say, oh, I'll use pressure. If I have access to enough pressure, I can change a fluid into a solid. And that's going along this axis, which is 1 over the density. So that's another way. And there's actually, in biological tissues, a third way, and I've given it a really weird name, and I will explain this later because this is new, and it's not very common in typical solid state material. So I'll come back to this one later in the talk. But let's discuss this one first, because this is one that we can talk about and understand in a fairly straightforward manner. Okay. So what happens when I go along this axis? And for this example, I can explain it to you most easily by thinking of a jar of marbles. Okay, so think of a jar of marbles and make them Teflon coated so they don't have any friction, because that's easier. So I have Teflon coated marbles. Okay, and our intuition is that the jar of marbles, if I have a big jar, the marbles can all move around and change neighbors, and as I compress the jar, all of a sudden they can't, and that's how they become rigid. Okay, so in the simplest model, there's an easy explanation. So if I have NP number of marbles, NP number of particles in D dimensions, so here D is 3, but you can do this generally, that's the number of degrees of freedom, because each particle can move in each of the dimension of directions, right? That's the number of degrees of freedom. And when two particles come into contact, that creates a constraint on their motion. So if I count the number of neighbors that each particle has, so if Z is the number of contacts per particle, then I can say, well, actually the number of constraints is the number of particles times the number of constraints per particle, but I should divide by 2 because each one of those bonds is shared by 2 particles, right? So this is the number of constraints. So I expect that the system should become rigid when exactly the number of degrees of freedom equals the number of constraints. And if I just set this equal to that, I get a simple expression which says that systems become rigid when the coordination, the number of contacts, is exactly equal to 2 times the number of dimensions, or 6 for marbles that are Teflon coated. So what happens as a function of density? Well, at low densities, I have less than this critical number of contacts, and so the system is what's called under-constrained. It has less constraints, okay? So it should be floppy. And then at high densities, I have more than this, and the system is over-constrained. And kind of, I would say magically, it works exactly. This works exactly for a jar of marbles if you go and do a careful experiment. It works. This mean field argument works exactly. So that's kind of cool. It actually turns out that both of these two examples I sort of highlighted in the beginning, this very careful work, look at this picture, this careful work is counting the number of contacts of each of the cells in this thing, very carefully, very good microscopy. And it turns out that both of these two examples have significant evidence that in these cases, the fluidization is caused by small changes to density and changes to the number of contacts. It's exactly this physics that's governing the behavior of these cells in development. That's so cool. Okay, so in these types of systems, thinking of cells as kind of squishy spheres is a really good approximation, and it works really well to explain the onset of rigidity. Okay, but I'm interested in epithelial cells as well. So epithelial cells are things like your skin that form a barrier function in your body, right? So they can't have holes and let anything through, okay? So they can't change their density. That would be really bad for you, right? If they change their density, they actually take up basically all of space to form that barrier function. So when I started thinking about this problem at Princeton. When I started thinking about this problem, it's quite clear immediately that these two mechanisms don't work. It doesn't work. The physics that I thought I understood doesn't work. Okay, so let's think about it. And to think about it, I'm going to go to an even simpler example. Okay, so let's revisit rigidity. And I'm going to talk about it even more carefully than I did before. You thought it was pedantic before. Now it's getting real. Okay, so this is these are nodes, and I've drawn them in 2 dimensions now. So this is a 2-dimensional set of vertices, and they're connected by rigid bars. And each one of those rigid bars is a constraint. So we're just going to count constraints and degrees of freedom again, right? Okay, so in 2D, I have 4 vertices, and that means I have 8 degrees of freedom. You see there are 4 bars here, good. It turns out that I need to know one more thing, which is I also want to forget about rigid body motions, right? So if I take this group of plaquette and I can either rotate it, that's one, or I can move it up or down. So I have to subtract those 3 rigid body motions in 2D. But once I do that, I see that I have one non-trivial floppy mode. And I've drawn it here, right? This is the floppy mode, this thing in the dashed lines. I can move the vertices that way, and they're allowed to do that. That's just constraint counting. It's pretty boring. And if you said, well, I don't want it to be floppy, then I say, well, just add one more constraint. Good job. You have now made it rigid, and yes, that object is rigid, and engineers have known this for an extremely long time, right? This is how bridges work. Okay, so now I'm showing you 2 plaquettes, and I want to introduce one more idea because it's important later. So if I have 2 of these squares, and I put the bonds in a smart place, I still get a rigid system. But what if I put the bonds in a dumb place, right? So I put both of my bonds in the same box and left this one floppy, okay? That would be, if I was trying to make something rigid, that would be dumb. But I can also count what's happening here, and it doesn't break my counting. And so the idea is that actually if I have 2 bonds, you note that that actually always introduces what's called a state of self-stress to my system. And a state of self-stress is basically a set of tensions on these bonds that leave the vertices unchanged. Okay? So I can put tensions. You can imagine I could put tensions on this object and it would still stay in exactly the same place. I could. Okay. So now we have to all change our counting. So before, we said that the number of degrees of freedom minus the number of constraints equals the number of 0 modes. But now I'm saying, well, actually, you have to count the number of states of self-stress, too, in case you did something dumb like that. Okay? So now we've done constraint counting again. Okay, but it turns out that this is still wrong. This is still wrong for a large class of materials, and I'm going to tell you why. But first, I'm going to give you the examples, because I think the examples are very interesting. Okay, so those fiber networks I talked about that are inside your body that are useful for you, they are almost always under-constrained. Okay, so let me tell you what that means. So you can approximate these fiber networks by a bunch of springs, and it's actually a really good approximation to their behavior, it turns out. And so you have an actual length of each spring and a rest length of each spring, and the energy is quadratic in that. Okay, so if I have a network this, you can kind of see that looks like a bunch of particles, that have interactions between them. And so it's the same exact counting. If I have a coordination that's less than 2D, then this system will be under-constrained. And actually, if you look at like collagen networks in biology, they really have coordination numbers that are 3 or 4. So they really are under-constrained. So they should be floppy. They should always be floppy, right, by this constraint counting. Okay, but what you find is that if you took, and I told you this already, but now I'm showing you it in a different way. If I take a box of my fiber network and I increase the size of the box, I deform the box, there's a critical point at which the system becomes rigid. So a certain amount of deformation at which the system becomes rigid. So if I want to think about it, if each spring had a rest length that's the size of this red bar, what I've done, basically, when I stretch the box, is I've made all the springs larger than the rest length, and they're all unhappy, okay? All those springs become unhappy, and this thing becomes rigid. Just because it's going to be important in a minute, I'm also going to say, well, actually, like if you think like a mathematician, then I could equally well, instead of changing the size of the box, I could keep the box fixed and just change the rest length, and that should be exactly the same, and it is. It's exactly the same. Okay. So what happens if I do an experiment on a collagen network, and I do this thing, I'm just going to show you some data. This is how stiff the network is, and note that this is a logarithmic scale. So this is real data on collagen networks. And this is the applied deformation to the box, so how much deformation I apply to the box. And so what you're seeing is like over here, the measurement of the stiffness is pretty low. I mean, it's something, but it's not very high. And then sort of around this black line, there's a critical amount of deformation where this sort of jumps up, and it looks kind of underwhelming, but this is a logarithmic scale, right? So this is jumping by orders of magnitude here at this critical point. So it's really not underwhelming for for actual cells. Okay, so this is just a picture. And I told you we would also talk about those biological cells that we discussed earlier too. So that was an easier example. Cells are harder, right? Like a spring network, I can imagine that I can represent it by like a little unit of springiness, and it might work good. But for cells, maybe not. However, there's this amazing, I would say, long body of work, starting by Honda quite a long time ago, that suggests that I can model cells in a very simple way in these cellularized networks. So the idea is that I basically treat each cell in one of these epithelia where they're all really tightly packed together and there's no space between them. I treat them basically as like almost like a foam, right? And I give each one of those interfaces an energy, and then I write down a mathematical model, and it's called a vertex model. For some statistical physicists, it's not the vertex model it's a different vertex model. I basically assume that I can say that the energy of one of these cells, or the mechanical work that's required to have a cell of a given shape, is related basically to its surface area, its volume, its perimeter. Those things that describe the polyhedra or the polygons in the tissue. Okay. And so this is the only equation I think in the whole talk, but I'm going to show one equation. I showed one equation about the springs, but that was a super easy one. Okay, so this is actually, if you look at it, a very similar equation. It basically has a P0, so that you can think about this kind of like the rest length of a spring. I'll tell you what it is in a second. But look, it looks very similar. There's a spring constant, and it's basically just the difference between the actual P of a cell and sort of the rest P of a cell, okay, squared, exactly like the springs. Okay, and what was discovered, I think, in these like lovely series of papers, and I would especially highlight this beautiful paper by Reza and others, that you can identify a shape index for cells that's really important for describing its mechanics. So the idea is if I say, well, I'm going to abstract one of these polygons, well, two things I might try to measure are its area and its perimeter, right? So this is a top-down view of an epithelial layer, and I'm looking at the outline of one of these cells, right? And if if I'm a dumb physicist and I try to describe it in some mathematical way, maybe I say, okay, the area and the perimeter are important. Now, there's a lot of things that I'm not going to talk about in this talk, and like years and years of work that suggests that this model has deep underpinnings in things like, well, it's even cut off in this picture, sorry, cadherins and myosin and molecules that are, that mean things. But I'm not going to go into the details of that for this talk in particular. What I will say is that you find that the key parameter for these models is this target shape factor, which is a dimensionless number based on this picture. And if I think that the area and the perimeter are important and perhaps independently controlled by cells, then I can make one dimensionless parameter, which is basically the perimeter divided by the square root of that cross-sectional area. So what it means, if I have a cell that's more rounded, it has a small perimeter relative to its area. And if I have a larger perimeter relative to its area, I get something that looks more like a pancake. Right? Okay, and so if I change the shape factor, what we discovered is that I actually undergo a fluid to solid transition in a model tissue. And I have to tell you, this surprised the heck out of us when we found it. We weren't expecting it. Because over here, when the cells have a more pancake-like shape, they're more elongated, the whole tissue, emergently, the tissue is a fluid. And then when they become more rounded up shape, the thing becomes a solid. Okay, and before you say, well, that's a stupid model, it is a stupid model. It is a stupid model. But the crazy thing is that it's actually highly predictive. So first let me say, okay, if we want to compare to an experiment, how might we do that? Well, if there's some, how I said there's sort of fluctuations, kind of like temperature in tissues. They're not quite temperature, but there's some fluctuations. So if I have a system that has some fluctuations, but not too much, and what too much is complicated, but let's just say we're there, then if I'm in a solid, I expect that those fluctuations aren't going to be enough to make the cells change their neighbors, right? A feature of a solid is that all of the molecules in a solid keep their neighbors, right? And so if I put a little temperature on that, nothing's going to change. But if I go to the fluid phase, then I expect even at that exact same temperature, if I'm in the fluid, then I'll see the cells all moving around and changing neighbors in the fluid phase. So that's something you can measure in an experiment. You can say, how often do cells change neighbors as a proxy for whether the cells are in the fluid state or the solid state, assuming that there's some small level of fluctuations in a tissue. And so actually working with Karen, who's right there in the audience, her research group, fantastic collaboration. We did this in the body of a fruit fly. So in fruit flies, there's also this body axis elongation process. And I'm not going to go through all the details today, but this over here is that shape index I talked about before. And this black line is actually a theoretical prediction with no fit parameters for how this transition point between a solid and the fluid depends both on the shape, and it turns out that in the fruit fly, you have really strongly anisotropic forces. So we also need an additional number to describe, okay, for those of you who are experts, a nematic order parameter. So you can think of this axis basically as a nematic order parameter if you want, but it's just an additional parameter of shape that we know how to measure in vertex models. And this black line, again, is a theoretical prediction a priori with no fit parameters. And then this data is from a fruit fly, actually lots of fruit fly embryos. Thank you, Karen. And the color is telling you how fast those cells are changing neighbors. Because of course, we get pictures like this, so we can measure the shape index, we can measure the nematic order, and then we can also measure how fast are cells that have that given set of shapes, how fast are they changing neighbors, okay? And so the blue stuff is when they're not changing neighbors up to our experimental accuracy. The orange stuff is when they're changing neighbors. I would say for a biology experiment, this is actually a really, quite frankly, surprising, remarkable agreement between a priori theoretical prediction and the behavior of like a live animal in a microscope. And so I'm just showing you this one plot. There's actually lots of work that we've done, but the reason that I'm showing you all of this is like this model works for epithelial tissues, and then we can try to understand a deeper question, which is if we accept that this model works to describe a living organism, a living epithelial tissue, why does that thing become rigid? Okay, like what the heck? Okay, so the question is what causes rigidity in these under constraint systems in the extracellular matrix and also in these cellularized models? And now we have two models that we think are pretty good, so we can think about it in terms of models. Okay. Okay, so now we revisit our picture again, and I want to say, okay, how is it possible that my constraint counting went wrong? This is like a very simple argument, right? How did it go wrong? Okay, so we're going to talk about how it went wrong. So remember this equation from earlier. It says I have to count the number of degrees of freedom and subtract the constraints. But this is actually a linearized theory that predicts this. And so under constrained systems generally don't have any states of self-stress, but in special geometries, in special configurations, they can appear. So let me show you what I mean by that with this simple example again. So this is our square, just as before. But now imagine that I'm allowed to change the geometry of this square however I want, and I choose to basically pull apart 1 and 4 so that they're almost exactly, so that the distance between 1 and 4 is almost exactly the same as the distance, is like 3 times the distance between the other junctions. So basically, can you see that this is going to make it almost a straight line when I do this? Okay? So it makes almost a straight line. So this thing, it turns out this thing, which I told you, it's under constraint, and we all agreed it's under constraint and floppy, it becomes rigid when I do this. Okay, so how do I understand that? Well, if I did constraint counting, I still have 8 degrees of freedom and 4 constraints. But now I've introduced a state of self-stress, and so when I subtract my 3 rigid body motions, I actually have 2 linear zero modes. And I'm drawing them here for you with these green arrows. So can everybody see that if I basically have a system that's a straight line, what this math is telling me is basically there's a perturbation of this 0.2 and this 0.3 that costs 0 energy to linear order. But of course, you can see by the geometry of this that they actually do obviously cost energy to second order. Okay, so where my constraint counting went wrong is that I need to include second order contributions to this. Okay. So this was already known. Okay, now the question is can I use this idea to understand how these more complicated non-symmetric, disordered structures in 2 dimensions and 3 dimensions, how they become rigid. And first order rigidity, so what is this picture? This picture is a Venn diagram of types of rigidity. Okay, and it turns out the kind that we care about is this energetic rigidity. It's basically like, can something flow or not? Can something move or not? And that's this big bin. And it turns out that constraint counting or first order rigidity is just it's necessary but not sufficient for energetic rigidity. So if something is first order rigid, it's rigid, but it doesn't guarantee, right? And so what you find is there's this whole hierarchy, and in this paper, recent paper, we go through it, there's this whole hierarchy of rigidities, and second order rigidity lives here. And I won't go through any of the details in a public lecture, but we proved in this paper, and it's a ton of math, so it's not in a public lecture, that all of the systems I talked about, spring networks, vertex models, different kinds of vertex models, they all do become rigid precisely due to second order rigidity. There's this geometric incompatibility that we understand, and it basically means, and this is kind of interesting, that, at least models of your tissues, they have an extensive number of first order floppy modes in them, but they're all rigid. All of those motions are constrained to second order in the perturbations. It's kind of interesting, and you wonder if biology can use it. But this was lovely because now we have a very deep understanding of how tissues become rigid. And, we can also start to think about how that might be useful for designing materials. It turns out that actually my collaborator Chris Santangelo showed that this is also how origami becomes rigid, is actually via second order rigidity. And you can make all sorts of very interesting mechanical metamaterials from origami, which means that what's a mechanical metamaterial? It's a material where the emergent properties of the object are quite different from the inherent behaviors of the object. So for origami, a piece of paper has a mechanical response if you try to stretch it that's pretty stiff. But if you think of a folded origami pattern, like if you've ever folded a crane, you pull on its wings, it has some stiffness, but it's very different from that of the paper. That's the idea. And people are starting to think about how you can actually use these type of second order rigid structures, because it also turns out they have bistable points that are useful for computing to make mechanical computers. And one of the things that we're thinking about doing is making sort of all kinds of designer materials. So it turns out that we can sort of parameterize the manifold of second order rigid states for like simple structures and more complex structures. And we can sort of design materials. I'm not going to say anything about this for the rest of the talk, but I think it's interesting that biology has inspired this idea of how to make materials that have these very interesting and programmable mechanical properties. Okay, so the conclusion of this part is that we mostly understand now, I would say, the physics of how macroscopic variables like density and cell shape and fluctuations govern emergent rigidity. Okay, so that was a difficult problem, but I think it's largely addressed. Okay, so in the last few minutes, yes, of my talk, I'd like to talk about challenge 2, which is how do you get a system to tune those variables robustly? Okay, and this is ongoing work, so it's a little bit more speculative. Okay, and what about these forces? You talked the whole time about these stupid material properties, but actually making the forces happen where they need to happen is actually very interesting, and you should try to do that too. Okay, so the idea I'd like to propose for this is the idea of physical learning. And what it means, and I'll explain in a minute, it means you take advantage of those many tunable degrees of freedom by thinking about those as variables that you can tune to learn to execute a task. Okay, and so I don't know if you've all seen this really cool set of work that's come out in like the last year or so, but it's so crazy. So this is a picture of basically a group of people that took a vibrating plate and made the vibrating plate learn to recognize numbers, digits. It did an image recognition task, okay? This is a resistor network. It's just a resistor network, and it's a very small resistor network. This resistor network can do an image recognition task, okay? And this is a mechanical transmission line that basically can learn to be in the configuration so it can transmit forces at nearly no energy loss. Okay, so these are the tasks that these objects can do. And the secret sauce is they all have internal tunable degrees of freedom that you can actually adjust in order to make the system do this. So in here, it's like the sound of an amplifier. Here, it's the variable resistors, so you can change the variance of the resistors in this network. And here, it's the length of these springs on the side. You can tune that in that model system. Okay, and so the idea is, well, I kind of think this might be just what we need in order to program changes to forces and material properties in tissues, because of course what we're trying to do is basically doing computation before there's neurons that have evolved. There's no neurons in these early embryos, but you need to execute these tasks and you need to do it with exquisite control, and I think it's a really interesting idea. Okay, so of course the challenge is, this is a picture, like a standard picture of a standard neural network, and the challenge is in neural networks, we use a process, maybe some of you are aware, called backpropagation in order to estimate the global gradient with respect to a cost function to execute a task. It's okay if that means nothing to you, but in case it does. We need to be able to do, we say we have an input, we have a desired output, we have to adjust the weights in this neural network in order to get the output we want, and the algorithm that most people use to do that is called backpropagation, okay? But of course, the question is how could tissues be performing backpropagation? How could we I said there were these tunable parameters inside tissues, but how could tissues program them, right? How can they do this backpropagation? Okay, and so I just want to say there's been this really nice, I would say, progress in theory, very recent, and it comes by a bunch of different names: equilibrium propagation, coupled learning, or frequency propagation, that if you actually have, that basically you can do it. So in physical systems, like this resistor network, if you have two channels, which means there's a physical channel with the physical degrees of freedom and a learning channel with the learning degrees of freedom, then it turns out that doing learning in the learning degrees of freedom is constrained by the physical degrees of freedom in such a way that it makes backpropagation local. And again, if that means something to you, it's a very cool idea, I think. And it means that tissues can probably do this because tissues have such local rules. So for example, it's known that if I have an extracellular matrix network and I have a spring that's under a lot of tension, it won't get cleaved by enzymes. But if it's not under a lot of tension, then it will. And you can think about that as a local learning rule on the tension in the spring, right? And there's similar ones that exist in cells too. I'm not going to go into the details, but for cellular systems, similar things exist. Okay, so in the next 2 minutes, I just want to show you like a first step in this thing. So the question is, okay, if I take one of those vertex models, for example, can I make the thing learn by tuning the degrees of freedom even in that mathematical model as a first step? Can I do it? Can I tune the local properties to optimize a cost function? And I'm going to just use a global gradient here, and I'll worry about locality later, doing it locally later. I'm going to skip this slide because I'm basically out of time. You can do something like this in particle packings, and it leads to really cool states. You can find really cool states. But the question we're going to ask is, can I do it in this vertex model? And remember, the degree of freedom that's tunable in these vertex models is the cell shape. And what we say is, oh, well, if I can actually tune the average cell shape, if I can move around the average cell shape, that's exactly what I need to do actually to go back and forth across the transition. So that's kind of cheating. Like if I can just change the global cell shape everywhere, then of course I can get a fluid-solid transition. But what if I just say, okay, the only thing I'm allowed to do with my local rule is sort of keep the distribution of cell shapes fixed. So each of these colors is a cell shape, a different cell shape. And this set of cell shapes is exactly the same as this set of cell shapes. I've just shuffled around where they are, right? So I can say, well, what if I play the game, or what if I want to ask, can I tune the rigidity of a system by shuffling cell shapes? And, this is a task that we hope generalizes to other learning tasks. And the answer is yes. So this is the stiffness of the tissue, okay? And this is how the stiffness of the tissue behaves as a function of the average cell state, which I showed you before. This is a solid-like behavior. This is a fluid-like behavior in black. And when I turn on learning or tuning in those learning degrees of freedom, I shift the transition. In other words, if I'm allowed to learn in the cell shapes, I can basically enhance fluidization in the tissue. So this is a kind of simple proof of principle that, at least in our mathematical models, it is possible for a tissue to tune its macroscopic properties via basically one of these physical learning rules. Okay, so I'm not going to discuss this. I'm going to say, okay, what's next? So in this space, if we want to start talking about forces, we can think of cost functions that induce forces and deformation. So the idea is actually I can do learning. I can tune my internal degrees of freedom, my cell shapes, to try to change the shape of my box from a square to a rectangle. And that I think that can be done, right? And also allosteric responses, which is how proteins work. You can actually change the shape. Basically, you can have big deformations of a protein with only a small enzyme activating it. And we think we can do stuff like that in tissues too. And then also we're interested in taking some of these examples that are already in the biology literature, like these local learning rules, and figure out what cost functions they're minimizing, right? Like what are these local rules at the cellular scale, if you think about them as a learning rule, what do they do emergently? I think that would be a really interesting direction. Okay, so in summary, Morphogenesis requires simultaneous control over applied forces, right, like that rod that the glassblower has, right, and material properties, changing the, the heat that the glassblower has to change the viscosity of the fluid. Biological tissues undergo a fluid to solid transition to facilitate these large-scale deformations, such as body axis elongation and a bunch of other stuff I didn't have time to discuss, but it shows up in a lot of different places. Okay, there are multiple pathways that tissues can use to regulate their fluidity. We talked about first-order rigidity, which is changes to density, second-order rigidity, which is this metamaterial-like behavior, and fluctuations, which we didn't emphasize too much today. The last point is that physical learning, I think, is an emerging paradigm that might be useful for explaining how organisms control these forces and material properties. And we have one small example where we demonstrate that biological tissues can tune their global fluidity by tuning basically this internal learning degree of freedom, which is the cell shape. Okay, I'll stop there. Thanks.
Wonderful. We have time for some questions. Just push the button.
Yep. Is that working now? Okay. Of course, my memory is not that great, but I think that d equals 2 is a lower critical dimension for some of these. So, but in epithelial cells, you have 2 plus epsilon dimension, right? Because you can, so that would suppress fluctuations. And I wonder if you can comment about the role of the epsilon in, real transitions.
Yeah, that's a great question. So even in, you know how I said that we still don't understand the physics of the glass transition? There's actually a lot of question of what's the upper critical dimension for the glass transition. And there's some evidence that by some measures, actually it's d equals 2 for that problem, and that it is actually d equals 6 for other aspects of the problem. So there's some ways in which even 2-dimensional and 3-dimensional glasses are below the upper critical dimension. So let me say that first. Then the second point is that in epithelial layers, this second order rigidity transition is really nice because it's not, so it's there, I keep calling it a critical manifold. So it's critical in the sense that like a susceptibility diverges, but it's not critical in the physicsy, phase transition-y sense. There's no RG there. It's actually much simpler in a lot of ways. So for the epithelial cells, actually we don't have to worry. I mean, it is true that there is a plus epsilon, and actually there's an interesting question there from a mechanical rigidity perspective of whether that third dimension needs to be included when you're thinking about the mechanics of that second order critical point, but not from an RG epsilon standpoint, sort of more generically. And so very briefly, we're working right now on stratified epithelia where there's basically this one layer at the bottom and then there's multiple layers on top. And that epsilon becomes hugely important because you start to have cells that migrate halfway up out of the basal layer and they migrate halfway up, and then that epsilon becomes really important for understanding whether the rigidity is governed by the 2-dimensional behavior of that basal layer. Like, a cell escaping to the third dimension, should that be governed by the fluidity, the 2-dimensional fluidity of the basal, or the 3-dimensional fluidity of the whole? Because it's kind of like a liquid crystal in the sense that it sort of has an ordered state in the bottom, and it's very interesting. Yeah.
Pneumatic transition in liquid crystals. I mean, is there a connection?
Yeah, well, I just said a little bit. So let me repeat the question. The question is the fluid-solid transition similar to the pneumatic transition? So the first thing is that, of course, the pneumatic transition for rods is completely, you can think about it as completely governed by entropy, right? Basically the entropy of the states. And a very interesting thing is everything I talked about for those epithelial layers, you can actually do it all perfectly fine at 0 temperature. So you can think about how entropy might influence, like basically thermal fluctuations influence the rigidity transition, but that transition happens even at 0 temperature. It's not driven by entropy at all. It's actually driven sort of by this mechanics I showed you and with the simple examples. And so there is an interesting question about systems that, because it turns out that actually a lot of biological tissues are pneumatically ordered. And I showed you that diagram for a 2-dimensional system where I had like, I don't know how to alter these slides, so I'm going to give you a human picture of the plot. Like on this axis was the pneumatic order, and on this axis was a shape parameter. And so what that plot told you is, yeah, if I have some pneumatic ordering of the cells, it can alter where the transition occurs in a way that we can predict analytically. So there is an interaction between pneumatic order in these systems and where the rigidity transition occurs, but the mechanism that drives the transition is different from what drives it in liquid crystals because it's not entropic.
Talk a little about memory in your study.
Yeah, that's a good question. So what I would say is that there's a couple of ways in which the idea of memory enters into what we're thinking about, especially in terms of physical learning in these systems. So the first way is that you may want to invent something that can do like universal computing, like a neural network. And if that's an ultimate goal, you also want to have systems that can encode memory and can store it. And so what we focused on so far is not trying to build, not trying to think about these tissues as universal computers. We're not trying to recapitulate neural networks. I mean, you actually might be able to, but that's not what we're trying to do first because we're envisioning that we're more task-oriented. So we don't have to be able to universally compute everything. We just have to optimize a specific task, and we might have systems that are optimized to do one thing instead of everything. But there is an interesting question of if you can have histerons or basically elements inside systems like this that could do memory storage. And the short answer is, I don't know. But I think it's an interesting question.
So Lisa, so in your work you describe the bulk properties of the material. But in embryos there are regions that have very distinct properties. So for example, when you have an extending germ band, you have a part of a material that flows and then 20 microns away from it, it will be solid. So do you have an understanding of how at the same time you span different parts of the diagram?
Yeah, that's a really, so like in particular, exactly, in fruit fly, there's the amnioserosa, which is sitting there, which is basically acting like a rubber band that helps pull as the tissue around it fluidizes. And so it's actually really important that part of it maintains elasticity. And so the short answer, actually there's a really cool paper by Christoph Schmidt and like Andrea Liu and people about the amnioserosa. And what they find, in that paper, very recent claims, is that the cells in that region are basically programming their cell shape to stay in the solid, even through these very large deformations, and that the cells in the germ band are executing a different program where their P0 is changing in order to fluidize them. There's a boundary between them that must have some surface properties to sort of insulate mechanically that. So I think it's a very good question. I don't know the answer of the mechanical insulation, but what I will say is it's sort of fairly straightforward to assume that when I have differentiated the cells that are going to form the germ band versus the amnioserosa, there's genetic control over those P0s that's different. So, I mean, what I would say is the way I think about it is like the bulk piece is maybe fairly straightforward. I think sort of how the mechanical insulation between those two parts is an open question and a good one. I don't know the answer.